Hello, everyone. Good evening. It is 7:17 p.m. on Friday. Hope everyone had a good weekend. A lot of things going on in the world. But today, what I want to talk about is one of the deliverables that we promised all of you from the newly launched CytoSolve Open Science Project. As many of you know, CytoSolve is a technology that I created out of my PhD work at MIT, which has evolved over the last 16 years to become a de facto platform, the leading platform in the world in systems biology for doing analysis of all sorts of biomolecular reactions in the body. And in fact, all different kinds of things we could use it for. We can use it for plants, we can use it for humans, we can use it for animals, but it eliminates the need for animal testing. And that's CytoSolve, and I'll talk about that. But a few weeks ago, in fact, it's been over probably the last several months. Many of you have implored me, hey, Dr. Shiva, can we use CytoSolve to look at the ingredients in the jab? Okay. And so what we did as a part of that was we said, we went to CytoSolve and we said, hey, look, can we start using the technology for supporting what I call open science? So this is the beginning of the CytoSolve Open Science Project. We're going to start doing many, many projects, and I'll do a whole video on this. But the concept of open science is that we do not wait for government to fund science. We know that the collusion of government and big corporations has become so tight that you can't really trust what comes out of many of the large institutions because of their linkages with big pharma. So it has been one of my privileges over the many, many years as a scientist to figure out how we can make science much more democratic. What's happened is over the last 15, 20 years, particularly starting with the Mansfield Amendment in the 1970s, where large amounts of money got moved to institutions like the NSF and NIH, science became highly politicized. And so, again, I'll do a whole video on this. And in 2003, when I created CytoSolve, CytoSolve was a breakthrough technology because it allowed us to look at all the research out there, and I'll show you this, bring it together without choosing the winners or the losers in a very objective way, and then mathematically model the molecular mechanisms. It's a way of distillation of science that we could do very complex analysis faster, cheaper, and better than the biggest institutions in the world. In fact, we just recently published a paper in the leading cancers journal where we mapped out all the molecular pathways of amyloid leukemia, which would have probably taken anyone else, any other major institution. So we have this revolutionary technology. So we want to make CytoSolve now accessible to the world, where every one of you can directly ask a question and directly get scientific questions answered. So as a first pass at this, many of you asked, hey, what is in the Pfizer Jab? And we got the actual data from the FDA. And then that's what they are saying in it, So we use that, and we're going to do a series of analysis using CytoSolve. First of all, what's in the jab? So we're going to today share with you the first step, what's in it from a chemical analysis. So you're going to learn that. Next, we're going to start running the ingredients. We're going to model, for example, the molecular pathways of inflammation. And we're going to start looking at how each of these ingredients affect it. We can do it for various things, but inflammation is a very profound thing. And then we're going to start looking at what happens in combinations. So this is going to be a research project by the people for the people. And so if anyone wants to support this, which, where many of you know, I've been extremely generous with my time and a lot of things, but we have to be careful because we don't want to burn out. So we need your resources. So you can donate $1, $2, $5, whatever you want. But this is going to be directly customer, consumer, citizen funded research. So when you donate, we're going to use that funds to support our scientists to use CytoSolve to actually find out. But we're going to be able to do all of this research at a fraction of the cost what anyone else can do because of the technology we have. So today's thing is to answer that question, to start answering that question, what's in the Pfizer jab? And more importantly, how can it actually affect individuals? So let me go right to this. So I've set this up. Again, we want to thank our research team because a lot of this our research team has to do actual research projects. So we literally take their time and we move it over here. And it's your funding that's made this possible. So again, the Open Science Project, as you can see in this slide, it's again, CytoSolve's in the upper right. It's part of our Truth, Freedom, and Health movement. And we're looking at the analysis of the Pfizer jab. This is part one. We're going to talk about what's in the jab, and it's a CytoSolve molecular systems analysis. You can see that if you want to know more, you can go to vashiva.com. But this is brought to you by the Truth, Freedom, and Health movement, and it is the Open Science Institute project. What this means is if you go to truthfreedomhealth.com, you can go right there and you can actually donate if you want to support the Open Science Project. And based on what you donate, we'll actually give you the Truth, Freedom, and Health platform too. So I never like to take anything for nothing. I want to actually support everyone's education. It's something my great-grandfather taught me. Education is really the ultimate revolution. So, and he was a poor village farmer. So let me jump right into it. So analysis of the Pfizer jab. So I want to welcome everyone. And it's part one of what's in the jab, and it's the Open Science Institute project. So by the way, if anyone wants to know more about what we do, all the different things we're doing, you can go to vashiva.com, and I encourage everyone to go there. So if you look at this, we need to first of all identify the Pfizer jabine. It's called Comirnaty, and there are 5 important bullets here that we want to go through. First of all, this is an mRNA based COVID vaccine. It's mRNA based, and the mRNA, as I've talked about earlier, messenger RNA is a piece of it's the half of DNA, messenger RNA, and the messenger RNA will code for a particular protein. Okay, so the mRNA-based jabs are really about a piece of mRNA which will code for the spike protein, and then your body will see that spike protein, thinks it's been infected, and the body will bring up an immune reaction. So that's what we're looking at here. Okay, hold on 1 second. Hey, Dominic. No, we're on our way. Sorry about that. Hey, John, let Michelle know that we have a dinner appointment. Tell her to come on over here with Dominic. Okay, ASAP. So sorry about that, everyone. She's on her way over here, but tell her that he's coming here. Okay. And then by the way, a friend of mine, we've been waiting to meet him for dinner, and I didn't know where he was, but it turns out in Boston, you have to get a jab to go somewhere, and we don't want to do that, so we're changing our restaurant plans. Okay, John, can you get Michelle to come over here ASAP? Thanks. So right here, what you see right here is that this vaccine was developed by Pfizer, and it was approved for individuals 5 years of age and older. And there's 2 doses in this: the primary series and the booster after 6 months for individuals 12 and older. Okay. So again, 2 doses you have to take, and the booster after 6 months for individuals 12 and older. So that's a background. And if you look here, I want everyone to look here so you understand sort of the history of this. Okay. So it's clear. What you'll see here is that in March of 2020 is when Pfizer and the BioNTech announced their partnership, March of 2020. And phase 1 and phase 2 trials began starting in April. So this was like Operation Warp Speed, right? They went right into trials, okay, clinical trials, and they started doing immediate commercial production. Look how fast they did it, right from mid-April right to July. The commercial trials start, right? Commercialization. It goes into commercial production. And then you have phase 2 and phase 3 safety, quote unquote, safety efficacy trials. And then the safety trial, the phase 3 trial is expanded to increase diversity of the population. But look how fast this is occurring. Typically, clinical trials take over many, many years, 5, 10 years. This is occurring in 1, 2, 3, 4, 5, 6, 7, 8 months, okay? And then they apply for emergency authorization. And so on August 23rd, 2021, this gets authorized. So this occurred in a very rapid time frame, from the time they started, let's say March to March, so within about 16 months, all right? Now what you see here is that there were many, many vaccines in development, right? There were 194 vaccines in development. They included nucleic acid vaccines, viral vector vaccines, protein-based vaccines, and whole virus vaccines. So 1, 2, 3, 4 kinds, and also the bacterial spore expression vector, but none of them are used here. But you can see ultimately what's out there right now is 3 of the viral vector vaccines, 3 of the nucleic acid, which is like the Moderna vaccine, 3 of the whole virus, and only 1 of the protein. So the whole virus vaccine is where there's literally a attenuated virus. You have a virus and you deaden it a little bit and you inject that into people. The protein-based vaccine is literally the spike protein. The viral vector is where you actually take another virus that's quote unquote not as harmful and you genetically modify it so it gets a spike protein on it. And the nucleic acid vaccine is the mRNA vaccine. Point is, there were many, many different kinds of vaccines that were taking place here. John, you got Michelle? Yeah, please call her, okay? Thank you. So that's what's going on here, all right? Now, what we see here is, what we're gonna learn is what is in the Pfizer jabine, what is the function of the Pfizer jabine, what is the concentration, and how are the Pfizer jabine components made, okay? So first of all, I want to emphasize that all of this would not be possible without the CytoSolve research team doing this. Again, it's the Open Science Project. You guys got to support this because we can't do it without your support. Otherwise, I'm going to go bankrupt. So go to truthfreedomhealth.com, contribute to this, and you can also become a warrior while you're there. But the CytoSolve technology, it allows us to do some very powerful things at cytosolve.com, but allows us to look at all the research that's out there, distill it, so we're not looking at any one research paper. And most importantly, right now, pharma companies typically go through this long process of development, new compound, they do in vitro, in vivo, all these processes. And the pharma companies, before the jabine came, vaccine for them, they're actually been losing a lot of money. In fact, as you see here, they used to spend lots and lots of money on R&D, and they were getting less and less new drugs approved. But the reality is that this process has been breaking down. And this is why pharma was so thankful to Trump that he did Operation Warp Speed. John, get ready with the video. So I want to just take a couple of minutes so all of you understand what is CytoSolve, and then we're going to go back into our findings. Go ahead, John. So John's going to play a little video of what is what is actually in the or how CytoSolve actually works. Okay, so let me go. In fact, John, do you have it? Okay, go ahead, John.
Who would have ever thought someone like me would invent email and create CytoSolve to revolutionize health for personalized and precision medicine, a system for delivering the right medicine for the right person at the right time? I was born a low-caste untouchable in India's caste system, a system of aristocracy, oppression, and racism. As a child, I observed my grandmother, a poor village farmer, practice Siddha, India's oldest system of medicine, to heal local villagers by observing their face, to know their unique constitution, to deliver a unique combination of foods, healing herbs, and massage. The caste system and her abilities to heal inspired me to understand the interconnectedness of all life. My name is Dr. Shiva Ayyadurai. I'm an MIT PhD, a Fulbright scholar, a scientist, technologist, and inventor. My family and I left India to come to America on my 7th birthday. As a 14-year-old, I began working as a full-time research fellow at Rutgers Medical School to unravel the mysteries of sudden infant death syndrome, SIDS, and created the world's first email system long before I ever heard of MIT. As I traverse academia over the next 3 decades, I observe self-serving academics, never solving real problems, writing grant after grant, competing for tenure, while diminishing real science and real scientists, pushing a reductionist science to destroy the scientific method. Like the blind men who never saw the whole elephant, but the parts, they delivered a dismembered view of reality. I observe Big Pharma use such reductionism, wasting billions year after year to fund research in test tubes, killing animals, and using the poor as guinea pigs for clinical testing to create products that even the FDA no longer allowed. Not only Big Pharma practiced this reductionism, but also the elites of Big Vitamin, Big Green, and Big New Age, with gurus and yogis, empowered by Hollywood celebrities, selling one supplement after another based on a cherry-picked science. All that changed in 2003 when the Human Genome Project ended, revealing that humans have the same number of genes, about 20,000, as that of a worm, giving rise to a systems biology. We realized that one-size-fits-all medicine was a failure. We realized their medicines were killing us, making today's generation's lifespan shorter than any previous generation. Obesity, heart disease, deaths from adverse reaction to drugs, confusion on what diet, what supplements, and who to believe is what they have delivered you. They push natural and organic products for your beauty and wellness, while their real solution is their plastic surgeons and Botox. We've been sold out. It's time for real science, a system science that interconnects the parts to discover truth, to know what really works, to get the health we need and deserve. This is why I created CytoSolve. CytoSolve is about truth, freedom, and health versus power, profit, and control. CytoSolve is a revolutionary technology integrating bioinformatics, computational biology, mathematical modeling, decentralization to reveal the truth. CytoSolve computes trillions of potential combinations of biomolecular interactions to discover what actually works based on the actual science. No reductionism, no cherry picking. CytoSolve's predictive modeling has been proven accurate time and time again, matching laboratory results. CytoSolve discovers synergistic combinations of compounds to maximize health and reduce toxicity. For example, we know curcumin from turmeric and resveratrol from red grapes alleviate inflammation, but how much should we combine? Current methods are hand-waving at best. Here, with CytoSolve, we first model the control condition with no curcumin and no resveratrol to simulate high inflammation with the cytokine level at 0.15 micromolar. Next, we add just 5 micromolars of curcumin. The inflammation drops to 0.05. Next, we use 5 micromolars of resveratrol, and the inflammation drops from 0.15 to 0.06. But when we combine curcumin and resveratrol of 3 micromolars of curcumin and 2 micromolars of resveratrol, inflammation drops from 0.15 to 0.03, far lower, nearly 200% less than just one compound alone. That's the synergy principle of system science. We've all had enough of their fake and reductionist science. They think we'll simply keep buying their marketing, their celebrities, and their products that can never truly heal us. We don't need them. Great things come when we integrate the best of things. It's our time. It's time we delivered solutions for ourselves. It's time for truth, freedom, and health. It's time for CytoSolve. Welcome.
All right, everyone. So I hope that was helpful. That is CytoSolve. So now that you understand CytoSolve, CytoSolve is this very powerful capability that allows us to do this kind of analysis. So let's go back here. So what's in the jabin? Okay. So just get it from here. It's Dominic. Okay. Sorry, everyone. We're dealing also. It's Friday night dinner plans. But anyway, so what you see here, this is in the Pfizer jabin. Okay. Let me walk you through this. So there are a total of 10 ingredients. Okay. So remember, the Pfizer jabin is what they inject in people is a lipid nanoparticle. And that lipid nanoparticle inside of it are different ingredients, but it's a lipid nanoparticle. And I want to walk you through that. So in that nanoparticle, what you see here is that there are various pieces here. Hold on one second. Hold on one second for everyone. I'm going to have to Can you have Michelle send the address, John? Okay. All right. So anyway, so what's in this is the following. So let's walk you through this, right? In the mRNA, so one of the key pieces is, if you notice here, there are all of these particles here, okay? There's 1, 2, 3, 4, 5, 6, plus these 4 particles. And in this are lipids, but most importantly, there's the mRNA, the messenger RNA right here, all right? And this messenger RNA is an important part of it. So let's walk through that. So messenger RNA is this blue arrow with these negatively charged particles. That's one of the things in there. And the messenger RNA looks like this. You have, this is what's called an RNA, which codes for the protein. And there are different pieces of the protein, the spike protein. And these are getting into the weeds here, but these are all the pieces. So the S1 domain here codes for the domain D, the A codes for domain A, the B codes for domain B, and then over here, the S2 domain codes for these different pieces, as you can see here, okay? The S2 domain codes for the CD domain down here, the HR1 domain here, the CH domain over here, and this purple thing, which is the FP domain over there. So there are these different components of the domain, okay? So those are the different pieces that are occurring in the Pfizer Jab, okay? So what's happening here is that when you look at this entire protein, it's got 1,255 amino acids, okay? That's the number of amino acids. So 4,284 nucleotides in the RNA codes for 1,255 amino acids. So that's this spike protein. Another way of looking at the spike protein is right here. And you can see it's about 17 nanometers in height and 10 nanometers in width. And to give you an understanding, this is very small. A human hair is about 100,000 nanometers. A nanometer is 1 times 10 to the power of minus 9, okay? And what we're seeing here is that here we see that mRNA 1 dose contains 30 micrograms, which is 0.003 grams. Total volume of 1 dose is 0.3 milliliters. The molecular weight of mRNA is 1,388 kilodalton. And to give you an idea, that means 1,388,000 grams per mole. And when you work out the calculations, and I had our science team do this, that if you look at the amount of mRNA in grams by molecular weight, what you end up finding is there's, in one jab, there's 12.96 trillion molecules of mRNA. So in one jab, there's 12.96 trillion molecules of mRNA. That's a big takeaway here. So from a molarity or a concentration standpoint, there's 72 nanomolars, okay? So ultimately what you see here is that's how much mRNA is there. It's 12.96 trillion molecules. What we see here now is that if you look at the lipid nanoparticle, it's composed of 4 kinds of lipids. The neutral lipid, which is right here, which is this blue, the ionizable lipid, which is a red, the PEG lipid, which is this, and the cholesterol, okay? And to give you an idea what lipids are, they look like this. They got a polar piece, right, which means it's attracted to like things like, water, and it's got the non-polar piece, okay, which means it will repel water. So the lipid is a macromolecule that's composed of a fatty acid. That's the fatty acid here. It's got the hydrophilic, the water-loving group up here, and the water-repelling group down here, okay? And when you look at it, there are 4 lipids in the Pfizer Jab: the PEG lipid, the ionizable lipid, the neutral lipid, and the cholesterol, okay? And the structure of the PEG, we're going to go through each of the 4, it looks like this, okay? And this provides a hydrophilic layer, which means it pushes away water to the nanoparticle, and it prolongs the half-life of the nanoparticle. So it's the thing that really supports the nanoparticle. Now, the other structure is the ionizable lipid, which is this piece right here, okay? Let me go back here. The ionizable lipid is the stuff here, the red particles. And the ionizable lipid provides, it's a neutral negative charge, and it stabilizes the mRNA, okay, inside that lipid. And then you have the structure of the neutral lipid. And this associates with the cholesterol with the nanoparticles and forms a core lipid structure of nanoparticles that mimics a cell membrane, okay? And then finally, you have the cholesterol piece, which is the one that's associated with the neutral lipid, and it forms a core lipid structure of the nanoparticles that mimics a cell membrane. That's what we got. So you have 4 lipid pieces here: PEG, ionizable, neutral, and cholesterol. And you can see the molecules here and the relative molarity of them, all right? Now, there are other ingredients, and there are many, many other ingredients. In fact, there's the ingredients that we're going to walk you through. One of the things I wanted to do was, before I do that, I want to emphasize to all of you that we have the basic ingredients, we have another set of ingredients. What I want to do, in the interest of time today, is, John, what do you think about we follow this up tomorrow? Yeah. What I'd like to do is, without rushing this, because of some of the events taking place, is I'm going to start this again tomorrow. We'll do this at the same time. But what we want to understand, what we just walked through, are the main particles of the nanoparticle from the CytoSolve Open Science Project. So all of you, we're going to continue this tomorrow, and I want all of you, to support this Open Science Project. It's just the beginning, and we'll do part 1 again. We'll start the whole thing again tomorrow. So I'm not rushing it, given that I have to take care of something personal, which we were waiting for someone and we have to rush. I thought we'd be able to squeeze it in when I was waiting for someone, but I'm rushing and I don't want to do that. Okay. So anyway, we will start this up again tomorrow, and it's the Open Science Project. Go to truthfreedomhealth.com. You can contribute, become a supporter, but we will start again tomorrow. Thank you.